Recording has been started? Okay. Okay, fine. So I'll just request you to send me the video link. Okay, the video of your recording video file. Hello. Yeah. 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 Okay. Okay. So in the chat box, I will write my email ID. So please copy that and please send the video on this email ID. Okay. Okay. Thank you very much. Yeah, so I've just sent my email ID in the chat box. So please copy that and send the video to me. Okay. Okay. Uh, okay. So we can start. So let me introducing myself. I am Chandra. So I am one of the teaching assistant of your course Cell Biology in the NPTEL platform. So, so in this course, I will mostly in today's session, I will try to discuss the assignment one of uh, previous course. Okay. So, yeah, somebody raise your hand. Dinesh Kumar, Maya Parmar. Yeah. So, any issue is there? Okay. Okay, so let me just start with the assignment week one assignment of the previous course of the I mean same course. So let me just present my screen. Okay. So guys, can you see the PPT? Is it visible?
Yes, sir.
Okay. Okay. So let's start about the with the assignment of the week one of previous course. So basically, in this session, we can divide this 2 hour session by two part. In one part, we can discuss the assignment questions of previous week or previous course, and in the other half, we can discuss your doubt. As you as you know that already there are some videos has been uploaded to the portal. So if you have any doubt regarding that, so you can ask me. Okay, so let's start with the first up. So this was the first question uh, uh, of the week one. So the first question is which of the following is a uh, general characteristic feature of the prokaryotes? Four options are there: presence of mitochondria, presence of nucleoid, presence of centrosome, and presence of Golgi body. So can so does so does anyone want to answer that or pick up any of the option?
Option
Can I
B.
have any answer?
Obviously,
Option
B.
option B. Okay, option B. Yeah, option B is the answer. So yeah, so uh, for that, if you just want to know about the nucleoid, so what do you think? What is the difference between the nucleus and nucleoid? Can anyone want to comment?
The nucleus is a double membranous structure. Nucleus is a primitive genetic material inside the prokaryotes.
Yeah, yeah. Which basically lacks the nuclear membrane, right? Yeah,
Yes.
it is a it is a primitive one. So yeah, so basically, if you just want to explain the concepts about the nucleate, as we know that nucleate is basically seen in the prokaryotes like E. coli. So this is a you know Can you see my can you see my cursor here? Hello, cursor is
Yes, sir.
visible here?
Yes, sir.
Okay,
It is visible.
okay, okay. So this is the typical circular E. coli genome. Here you can see. So as we know that the genetic material of the E. coli, the DNA of uh, the basically the E. coli genome is circular, and it has these different parts which have been colored with different color, right? So one of the part is the origin of replication, as you already know, right, left, non-structural right, and a replication termination uh, sequence. So basically, inside the host cell, inside the host E. coli organism, this genetic material, this genomic DNA is formed, is structurally organized. It is compact here. Uh, as you can see, and this is the in vivo genome organization of a prokaryote, specifically here, E. coli. So you can see that if we will follow the different color codes, so we can see that this termination region is here, compacted in here, this left region here, right region here, and this non-structural left is here, non-structural right is here, and the origin of the and the Oriwala portion of this E. coli genome is compacted here. And this green region, Ori region, contains the origin of replication, which is, as you know, that which is recognized by several replication starting proteins, and which actually cause the starting of the initiation of the replication. And here, the replication termination sequence, they are so this portion are, so this six portion, as we already divided the E. coli genome in these six portions, so these six portion is structurally organized in this way, and this entire thing is known as nucleoid. And uh, as I already told you, that in contrast to nucleus of the eukaryote cell, it is not surrounded by nuclear membrane. And the chromosome of the prokaryote is circular, and the nucleate forms by condensation and functional arrangement with the help of chromosomal architectural protein and RNA molecules. Yeah. So this is basically the structural organization of the genetic material in prokaryote. And yeah, uh, and this entire thing is known as nucleoid, which lacks the nuclear membrane, unlike the eukaryotic cell. So yeah, so this was explanation of the first question. So does anyone have any doubt in this question or in this? Okay, so I don't think there is any more doubt. So let's moving forward for the question two. So the question two is asking that the formation of acetyl CoA occurs in which region of the mitochondria? Options are inner membrane, outer membrane, Christie space, and matrix. So can anyone want to comment for that? For the right answer?
Sir, in the matrix.
In the matrix. Okay. Yeah, in the matrix. Matrix is the right answer. So if you just again want to go for finding or recapitulating the explanation for that, the conversion of acetyl CoA from pyruvate. So can anyone tell me the name of the enzyme which mediates the role of converting pyruvate to acetyl CoA? This is a complex, right? Many enzymes are there which mediates this conversion from pyruvate to acetyl CoA. So can anyone want to comment the name of the complex which plays this role?
Pyruvate dehydrogenase.
A dehydrogenase
Yes, sir.
complex. Yeah. So this is basically composed of,
Pyruvate dehydrogenase.
yes, pyruvate dehydrogenase, which is a part of the PDC, pyruvate dehydrogenase complex. Yeah. So this is the this is the explanation for how this pyruvate to acetyl CoA conversion is occurring here. So as you already know that in the cytosol glycolysis is taking place. So in the cytosol glucose, so the as you know that glycolysis will start from glucose. So from glucose, 6 carbon glucose will be broken down to two molecules of 3 carbon uh, pyruvate. So in this, after converting into the pyruvate, so there are of course many fate of the pyruvate, but in context of this question, the pyruvate will be transported to the uh, mitochondrial mitochondrial matrix. So here I have one question that mitochondria is double membrane organelle or single membrane organelle?
Double membranous structure.
Okay, double
Double
membrane.
membrane.
Okay, so what are the name of the membranes of the mitochondria? Can anyone tell me? Or how is being referred in the textbooks? The two membrane of the mitochondria. Can anyone tell me? One is outer membrane, which faces the cytosol, and another is inner membrane, right? Is it true?
Yes, sir.
Yeah. And the space between the outer membrane and inner, and the space between the outer membrane and inner membrane, what is it called? The space?
Intermembranous space.
Yeah,
Intra.
intermembrane. Yeah, yeah, yeah. So see here, if the pyruvate wants to go to the matrix, then it has to pass these two barriers, outer membrane and inner membrane. So after being transported to the mitochondrial matrix, this PDH, the location of PDH. Acha, what can be the localization of the PDH here? I mean, outside the mitochondria or inside the mitochondria? As someone told that the enzyme which mediates the conversion of
Sir,
pyruvate to
inside,
acetyl-CoA.
because
Huh? Sorry? Hello? Someone who's trying to speak something? Can you continue, please? Hello? Am I audible? Hello?
Yes, sir.
Yeah, so any yeah, so any uh, yeah, so any comment for that? What can be the localization of the PDH enzyme, pyruvate dehydrogenase?
Matrix.
Yeah, so inside the mitochondria, right? Then only it can have the access of the substrate, which is pyruvate. So can anyone tell me that in this conversion of the pyruvate to acetyl CoA, what kind of reaction it is? What do you think? What kind of reaction it is? Condensation, decarboxylation, oxidation, reduction. What is this?
sir, decarboxylation and oxidation.
Decarboxylation. Yes, decarboxylation. Because pyruvate is a CH3CO, then C double O minus, right? So here once CO2 will be removed and it will be from 3C, from 3C pyruvate, it will be converted into 2C acetyl CoA, right? Yeah. Uh, and one more question here, just I want to know your opinion in this regard. That what is the objective of the cell to add this CoA group to the acetyl group? this coenzyme group, this CoA, what is the objective of the cell? Why cell does want to do that, to attach one CoA group to this acetyl CH3CO group? So can anyone want to comment in this question, or do you have any opinion, any of you?
No, sir.
Okay. See, after addition of the CoA group, this molecule will be unstable, right? Right. It will be highly energetic. So it will contain high energetic bond. So now after getting this unstable, after getting unstable, so this acetyl CoA is very unstable. So now there is a possibility of acetyl CoA can take part in this TCA cycle, and this highly the you know because as this because low stability means high energy, right? So now as a unstable molecule, if it will be broken down through this TCA cycle, then as there is high energetic bond, so now there is a possibility of release of energy, which is in a form of what? ATP, right? Right. In the TCA
Yes, sir.
cycle, yeah. So in the TCA cycle, what are the mainly byproducts of the TCA cycle? One is ATP. Anything more?
NADPH and FADH.
NADP.
Not NADP, NADH and FADH2, right? Do you agree with that?
Yes, sir.
Yes, sir.
Yes. Yes. So see, in the TCA cycle, if you will see the byproducts of the TCA cycle, which is ATP. So ATP is a source of energy. Then NADH, FETH2. Acha, so ATP we can understand. ATP has a high uh, bond energy, so it can give energy to the cell. But how this FADH2 and how this NADH? So how can they contribute to the uh, actually, how can they contribute to give energy to the cell? This NADH or FADH2
So during ETS cycle, they help in generating
Through
ATP.
ETS.
in ETC cycle? What? Sorry,
ETS cycle, sir, in mitochondria.
yeah, I mean this electron transport chain you were talking about, right?
Yes, sir.
Hello.
Yes, sir.
Yeah, right. So NADH will. So yeah, so NADH in the initiation of the electron transport chain, which starts from the complex one. So from the complex one, NADH lose electron, release electron, and this electron will be transported from complex one, two, three, 4, and finally it will be accepted by oxygen, right? So in this process, when the electron will be transported through the inner membrane, complex 1, complex 2, complex 3, complex 4. So after due to this effect of this electron transport, there will be a formation of the proton gradient uh, across the matrix and the inner membrane space. And due to this formation of this proton gradient, the ATP synthase will be able to form ATP from ADP and phosphate, right? So in this way, it can contribute to form ATP. Same here for FADH2. Also, FADH2 will release 2 electron. NADH will release 1 electron. So their role is to release electron and to become oxidized. So because if they will release electron, then only the ATP synthase will be able to form ATP. So this is so in this way, indirectly by formation of the ATP, they are playing the role in in contributing energy to the cell. Okay, so yeah, so this was the answer that this conversion pyruvate to acetyl CoA is being taken place in matrix of the mitochondria. Okay. So next question was, in the experiment performed by Avery, McLeod, and McCarthy, the transforming principle was identified as what? DNA, RNA, protein, lipid, carbohydrates. So does
Option A.
DNA.
Option what?
A.
Option A. Okay, option A. Yes, option A is the correct. Okay, so now, so for this time, can anyone want to explain the experiment, which was done by Avadi, McLeod, and McCarthy, and how they found that this transforming principle was DNA, not RNA, protein, lipid, carbohydrate. So, what did they actually do? Can anyone want to explain?
That they use RNAs, protease, and DNAs, and
Yes, very good.
then they show the viability of the cell. And when DNA is put, then the
Oh,
cell dies. So they found that transforming principle is DNA.
okay. Yeah. So actually, when they added the DNAs to remove all the DNA, then DNA and after removal of the DNA, when they provided the you know S chain, then they saw that that there is no transformation of the S chain, and the strain of the S chain, which is Staphylococcus pneumoniae smooth, so they were remained as the non-virulent, right? So that's why they found that. But in any other cases, like when they use the proteinase to remove all the protein, or the lipase to remove all the lipids, so in those cases, they were found to say that the transformation was happened, and the successful transformation of A strain to the virulent R strain. So here, this is the actual experiment that was done by them. So here, first they have taken the Staphylococcus pneumoniae A strain, which is non-virulent. No, sorry, A strain, which is virulent, and they infected the mice with the virulent S chain, and the mouse was found to be killed. And similarly, when they infect the mouse with the art strain, which is non-virulent, so in the art strain, the mouse was kept alive. So yeah, but when so when they have taken the art strain, which is non-virulent, and the heat and the selfie extract of the S chain, which contains protein, DNA, RNA, carbohydrate, lipids, everything. So even there is no alive S chain, either there was heat kill S chain or the selfie extract of the S chain. So and the mouse was found to be dead, even in the presence of the art stand. So that's how the first time it was, the idea was there that there is something, transformation of the some cellular thing of strain extract was occurring place in the art strain, and as a result the mouse was found to die. So then there are the possibility as RNA, protein, DNA, carbohydrate, and lipids. So when they transform only RNA, so the non-virulent arsen was found to be non-virulent. When they transformed only the protein, the art protein, so the uh, arsen was arsen. And the same result for the carbohydrate and the lipid. Arsen was arsen. But when there is DNA in the cell-free extract. So in the presence of the DNA, they have found that there is transformation of the R cell to the non-virulent R strain to the S strain, and they was found to kill the mouse when they were used to infect the mice. So in these four cases, RNA, protein, carbohydrate, lipids, here in these four cases, no DNS was used. So as they have not used any DNAs, so here in these four cases, so DNA was there. So in the no, sorry. So here actually when they have not used the DNAs, when there is DNA, so in the presence of DNA, only the transformation of the non-virulent strain to virulent strain was happened. So that's how they found the conclusion that the transforming principle is DNA, not RNA, protein, or carbohydrate or lipids. Yeah. So moving on to the question 4. Which of the following organelle does not contain DNA? Nucleus, lysosome, mitochondria, and chloroplast. So yeah, so anyone comment to answer for that?
Option B, sir.
Lysosome.
Option B. Okay, option B. So nucleus has DNA, right? Mitochondria has mitochondrial DNA. Chloroplast has yeah, chloroplastidial DNA. So uh, yeah. So what do you think that what is the shape of the mitochondrial DNA? It is linear or circular? What?
Circular.
Circular. And in chloroplast?
Sir, chloroplast DNA is also circular.
Yeah, yeah. So yeah, so basically we can now actually have a resemblance of mitochondria with bacteria, right? Or prokaryote, because they have also the circular DNA. Mitochondria has also the circular DNA. So what do you think? Why that is? See other organelle like endoplasmic reticulum, ribosome, lysosome. So they don't have DNA. So why mitochondria needs this genetic material, unlike the other organelle? What can be the reason?
The endosymbiotic theory.
Sorry.
Endosymbiotic theory. They are prokaryotes and they used to be live freely.
Huh. Yes. Yeah, and also there is some mitochondrial proteins are there which cannot be synthesis, which actually needs the mitochondrial DNA for the encoding of those proteins. So for that, this genetic information is embedded only in the mitochondrial DNA, not in the nuclear DNA. Yeah, so that's why the mitochondria or the chloroplast they have the requirement of DNA, and also as you told that as they resemble to the uh, prokaryote, so that's why the genetic material is there, and that makes them special organelle. So yeah, so the answer is lysosome. So yeah, so as you already know that lysosome has acidic pH, right? Acidic pH of about 5. The inside of the organelle, inside of the lysosome, the pH is 5, and that is maintained within the lysosome. And lysosome has acid hydrolases, which can play role in the breaking down of the macromolecules like proteins. Yeah. So how? Yeah. So what do you think that uh, if the in some cases if lysosome will be busted out, okay, so and every interior thing of the lysosome, every content of the lysosome will be uh, out to the cytosol, okay, so then if this happens, then this acid hydrolase will also be in the cytosolic, right? So in these cases, if this happens inside the cell, so what do you think that can the other proteins will be broken down or what? If the acid hydrolysis will be leaked out to the cytosol, so the other cytosolic proteins, so will they be degraded? Will they be broken down or what? Or not? Can anyone have any idea? Can anyone want to comment for that? If it will happen, then what will be the fate for the
Will
cytosolic
it go for apoptosis?
apoptosis? No, I'm just asking that if acid hydrolase will be leaked out to the cytosol, then what will be the fate of the cytosolic proteins? Will acid hydrolase will be able to break down them or not?
Sir, in cytosol there will be neutral pH, so it will be
Yes.
inactive.
Yes, very good. So the acid hydrolysis is only active at the acidic pH, right? Which is only maintained inside the lysosome. Okay. And so, what do you think that what is the mechanism or what is the strategy for the lysosome to maintain the acidic pH, which is unlike from the cytosol, because cytosol has 7.4 neutral pH, right? So how can lysosome can
Through active transport of proton.
yes, yes, so which can pump the proton from the cytosol to the lysosome, right? And yeah, and these proton pumps are located in the lysosomal membrane, which actively transports protons into the lysosome from yeah from the cytosol, right? Yes. Okay. So I think all of you know that the proteins which are needed to be degraded, or the proteins which are localized in the lysosome, so they need to be transported from the to the lysosome, right? So so can anyone just briefly tell me that what is the transport procedure? I mean, from which organelle the proteins are being destined for the cytosol, destined for the lysosome? First,
From
the
the ER and Golgi.
yeah, air and Golgi. Basically, from the I think the outer layer is called the trans Golgi, right? So from there they will be yeah. So from there they will be transported to the lysosome. Yes. Okay. So which of the following combinations make a nucleotide? Four options are sugar, nitrogen base, phosphate group, sugar, phosphorus base, phosphate groups, sugar, nitrogenous base, calcium groups, nitrogenous base, phosphate groups. So, can anyone want to comment for the right answer?
Sugar, nitrogenous base, and phosphate groups.
Option
The
A.
first option.
Yes, sir.
Okay, so option A is correct. Yeah, option A is correct. Sugar, nitrogenous base, and phosphate group, right? Okay. Yeah. Yeah, so this is basically the structure for nucleotides. So they are deoxyribonucleotides or ribonucleotide. These four uh, images or figures.
RNA, sir.
They are sorry.
RNA.
They are deoxy, right? See, in the 2' position there is no OH, there is H, right?
Yes.
Yes, yes. So, so this is actually the they are deoxyribonucleotides. So they have this nitrogen base. This is adenine, right? This is adenine. This is guanine. This is thymine, and this is cytosine. So here the four. So the three parts of the three building blocks of the nucleotide: nitrogen base, sugar ring, pentose sugar ring, and there is a phosphate group. So as you can see here that in the 2' position there is H in the deoxyribonucleotides, but unlike that in the ribonucleotide there is OH, right? In the 2' position, which is not shown here. So now tell me if I will if I will apply base to this 4 nucleotide which are deoxy, and in addition I will if I will apply base to ribonucleotide. Okay, base means any alkaline solution like potassium hydroxide. So in the basic solution, what will be fed for this DNA and I mean this deoxyribonucleotide and so see DNA is made up of deoxyribonucleotide, right? And RNA is made up of ribonucleotide. So if a DNA will phase the basic solution, then what will be the fate? And if the RNA will phase the basic solution, then among the DNA and RNA, which will be degraded? What do you think? DNA is made of deoxyribonucleotide and RNA is made up of ribonucleotide. So in basic solution, which will be vulnerable for degrading? Can anyone want to comment?
DNA.
DNA will be degraded.
It will be single stranded. The hydrogen bonds will break.
Sorry,
The hydrogen bonds will
sorry.
break.
Hydrogen bond of whom? DNA or RNA?
So DNA.
Basic. No, because you see here when a base will attack. So for the attacking, so for the nucleophilic attack of the base, there is a requirement of 2' OH group, which is absent in the DNA. Okay, okay. So if there is 2' OH group, then only the DNA then only the nucleic acid, it can be DNA or RNA. So if it contains the 2' OH group, then only it will be vulnerable for the alkaline hydrolysis. So as the RNA has this 2' OH group, but DNA does not have, because deoxyribonucleotide does not have 2' OH group. Only ribonucleotide have the 2' OH group. So this basic hydrolysis will only applicable for the RNA. Okay, so the DNA will be safe. DNA will be safe, but uh, RNA will be degraded. So when you isolate DNA or isolate RNA, so in the isolation of DNA, what do you think? What is the, or if I want to store DNA, so the storing solution, the what can be the storage pH for the DNA? Should it be acidic or basic? According to the fact I told you, that in the basic solution DNA is safe, RNA will be degraded. So yeah. So what should be the storage pH of DNA and what should be the storage pH of RNA?
So basic for DNA.
Basic, slightly basic, right? 7.5 to 8. And for RNA, for RNA, yeah, for RNA, can the storage pH be basic, more than 7? Can be, can it be basic?
No, sir.
Yeah. So the pH should be slightly acidic. Basically, 5.2 is the optimized generally pH for storage of RNA. And also in RNA, as you already know, that the another factor of concern is the RNAs. So you have to store your RNA in the DEPC treated water where the RNA inhibitor are present, and also EDTA should be there. So yeah. So but the pH should be slightly acidic. 5.2 is the optimized. So you can't keep RNA in basic pH or more than 7 pH. Yeah. So moving on to the next question. The catabolic and anabolic pathways can be coupled in cells because the free energy released by one pathway can drive the other. Both the pathways are driven by the same enzymes. Both the pathways have same initial pH and same time cutoff. What can be the proper or the right answer for that? Can anyone want to comment for that? Hello, am I audible? Hello. Yes. Can anyone want to comment? Hello.
हेलो सर।
Yes, yes.
सर आपने असाइनमेंट दे दिया है?
Huh?
असाइनमेंट हो गया है कि अभी बाकी है ये असाइनमेंट?
It is going. Hello.
ओके सर।
I'm just discussing the previous year's assignment, not the current year's assignment. Previous years. So, yeah.
तो अभी तो बाकी है ना सर असाइनमेंट
Yes, yes.
अपलोड होने में? ओके।
Hmm. Okay, so anyone want to comment? Yeah. Okay, so the answer is the free energy released by one pathway can drive the other. So the simple example is ATP is formed from glycolysis and TCA cycle, and this ATP is uh, used to the biogenesis of many macromolecules. So in this way, as we know that the biogenesis of macromolecule is known as anabolism, and degradation of the macromolecules are known as catabolism. So that's how this free energy released by one pathway can drive the other. Free energy in terms of ATP. Yeah. So moving on to the next question. In the cell membrane, while lipids are arranged in a bilayer, which of the following statements hold true? So can anyone want to answer for that? Lipid arrangement in the lipid bilayer of cell membrane?
Sir, I think it's the second option,
Sir,
hydrophobic,
option
Hydrophobic head, then polar tail.
hydrophobic tail, then polar head.
No, it's not
no, sir, the
correct.
polar tail will be outward. Otherwise,
Polar,
because sir, the polar tail will be outwards. Because
yeah, polar head
hydrophobic will
actually,
be
right?
innerwards.
Yes, yes, very good. Yes, very good. Because the outer
Yes.
outer portion of the cell is polar, inner portion of the cell is polar, cytosolic is polar, but the membrane is hydrophobic, right? So that's how the orientation should be the option A, right? So the polar head, hydrophobic tail, then polar head. Okay. So this is the so now it will be more clear the lipid bilayer organization of the cell membrane. So here you can see this is the polar head in the inner membrane. So you can see that bilayer means two layer, like right in the inner layer the polar head group, as you told correctly, the polar head group is facing the polar cytosol, and in the outer layer the polar head group is facing the outside of the cell, which is also polar. And this tail, which are hydrophobic, which are the basically the fatty acid chains, they are very hydrophobic aliphatic fatty acid chain, so they are facing in the towards each other by hydrophobic interaction, and that's why this region becomes hydrophobic. So this is the orientation of the lipid bilayer of cell membrane. Yeah. So moving on to the question 8. The major difference in the cell wall structure between gram positive and gram negative bacteria is presence of a thick peptidoglycan layer in the gram positive bacteria, lipid bilayer in gram positive bacteria, thick peptidoglycan layer in the gram negative bacteria, and absence of cell wall. So which can be the true?
Option A.
Option A. Nice. Option A is correct. Presence of a thick, because the
Option A.
yes, 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 because the gram positive bacteria has thicker peptidoglycan layer than the gram negative bacteria. And here you can see the image of this is the gram negative bacterial cell wall structure. This is the gram positive bacterial cell wall structure, So in the construction of the gram-negative bacteria cell wall, you can see this is the plasma membrane. So in addition to that, there is a peptidoglycan layer which is thin, not thick as the gram-positive bacteria. So there is a thin peptidoglycan layer. Over that, there is a extra outer membrane, which 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 is composed by the lipopolysaccharide chain, purine, lipid. So this outer membrane is present in gram-negative bacterial cell wall, but not not in gram-positive bacterial cell wall. And here, if you will see the construction of the gram-positive bacterial cell wall, you can see there is plasma membrane. Then there is the gap between peptidoglycan layer and plasma membrane, which is known as periplasmic space, and there is a more thicker. You can see the peptidoglycan layer of gram-positive bacterial cell wall with respect to the gram-negative bacterial cell wall. So we can just see by this eye difference that the thickness of peptidoglycan layer is very high here, and it also contains the teichoic acid, lipotecoic acid here. So that's why the first option is correct, that the gram-positive bacteria has the thicker peptidoglycan in their cell wall with respect to the gram-negative bacteria. Okay, someone is waiting outside, so I should. Yeah. Yeah. So now, question 9. Pick the correct statement regarding the integral membrane protein. These are not transmembrane proteins. The hydrophilic side is exposed in between the lipid bilayer, may have one or more membrane spanning alpha helical region, have exposed hydrophobic structure on either side of the membrane. So which one is the true option regarding the integral membrane protein?
I think C.
Option C. Nice. Option C is correct. May have one or more membrane spanning alpha helical region. So what can you guess? What is the nature of this amino acid in the alpha helix of this membrane spanning alpha helix? So they are hydrophilic or hydrophobic? With the nature of this
So
alpha
they are
helical
hydrophilic.
hydrophilic. No, how can they be hydrophilic?
They can be present
Because
inside the cytosol and also outside of the.
no membrane. See,
Oh no,
membrane
sir. They. Sorry.
Yes, it is membrane spanning, right? So it is within the membrane, so it cannot be hydrophilic, right? Yeah, so it is basically hydrophobic because it is membrane spanning. It is present in the membrane, so it is hydrophobic. So the composition of the whatever amino acids are there in the alpha helical region, so they should be hydrophobic in the integral membrane proteins. So this is the basic, there are two kinds of membrane proteins, integral and peripheral. Uh, so what is the difference between the integral membrane protein and the peripheral membrane protein? So just by seeing this diagram, what do you think? What will be the difference between the integral and peripheral protein? Can you so are you able to visualize the difference? Any difference here? Integral and peripheral protein? Can anyone please come and want to say something? Just yeah.
Yes. Uh, the integral membrane proteins are present across the two
Yeah.
lipid bilayers, whereas peripheral protein is present only on the surface.
Yeah. They are basically not spanning the membrane, right? They are not spanning. They are not inside the membrane, right? Yeah. Yes. Yes. Exactly. Perfect. So this is the major difference between
Yes,
the integral and peripheral proteins. So in the integral proteins, integral membrane they has to have some transmembrane alpha helices which contains or transmembrane, or the shape can be the transmembrane alpha helices here, or some membrane protein also can have this beta barrel shape, where there is a lot of beta sheet which will make a barrel like shape, a basket like shape, a barrel like shape. So these two different kind of membrane integral membrane protein can be there. Alpha helical, which is uh, which has alpha helical region, and which has beta barrel shape. So these are two possibilities of having integral membrane proteins here. Also there can be some integral membrane proteins which are anchored to the lipid. As you know that the cell membrane has is a space for many lipids. So here specifically GPI is a glycolipid. So there is some GPI anchored protein. There is a protein. This protein is making a covalent bond with this GPI, which is a glycolipid, which resides in the plasma membrane. So, and in the case of peripheral protein, they either interacts with the integral protein. This is a integral protein, and this is a peripheral protein. So this peripheral protein is interacting with this GPI by non-covalent interaction. Or this peripheral protein also can be in a position where they can interact to the phospholipid of the plasma membrane by non-covalent interaction. Yeah. So these are two types of membrane proteins. Let me just see if anyone is waiting here. Yeah. So moving on to the last question. Where is the electron transport chain present? Cytoplasm, inner membrane of mitochondria, outer membrane of mitochondria, and endoplasmic reticulum. So what can be the right answer for that? Can anyone want to say?
Inner membrane.
Inner membrane, right? So option B is correct. You are telling yes. Inner membrane of the mitochondria. Their electron transport chain is present. So this is So can anyone want to explain this figure? This is the inner membrane of a mitochondria. This is a mitochondrial matrix. This is inner membrane. This is intermembrane space, and this is outer membrane, which we have already discussed in some earlier question. And in you can see in the inner membrane proteins there are complex 1, 2, 3, 4. So can anyone want to just tell us that how this electron transport chain is occurring here, and what is the objective? How can we get this ATP here? What is the motive of this of formation of this electron transport chain here? So can anyone just have a wild guess, or just want to say something? Can anyone come forward for that?
Sir, the electron transfer chain here helps to convert NADPH and FADH into ATP. Here the complex 1, 3 and 4, these three complex involve to push or pump the hydrogen ion from mitochondrial
Yes.
matrix to intermembrane space. And when those hydrogen ion return back through ATP synthase, it produces ATP. And for FADH, complex 2, complex 3 and complex 4 are used. But
Yes.
complex 2 do not push the hydrogen ion into the intermembrane space. So
Yes.
that is like point difference between ATP in
Yes.
NDH and FADH.
Very nice. Very nice. Yes, that was a key point here, of course. And here it is NADH, not NADPH here. Okay. So in ETC it is NADH, so it will just release two electron. Similarly, FAD here FADH2 will release two electron to form FAD, and these two electrons will be flowed, right? And due to this flow of this electron throughout this complex of inner membrane, the protons, as you told, from matrix to the proton will be so see here it is a concentration gradient, because gradient is, if you can see the arrow, the direction of the gradient is from mitochondrial matrix to the intermembrane space. So there is no energy, or there is no requirement of any active transport of transporting this proton from here to here, because there is already a concentration gradient where the concentration is more here and less here. So that's why this gradient will push the proton from the matrix to the intermembrane space, so that the concentration balance, so that the concentration of proton throughout the membrane space and the matrix will be balanced. So that is the reason of just transporting these protons here. So this is not pumping, but here you can see when these protons will come to the direction opposite to the concentration gradient, which is from inner membrane space to the matrix. So in this case, now it is just opposite to the concentration concentration gradient. So this kind of transport cannot be occurred by the simple this concentration gradient wala process. So for that's why it is a so for that's why this direction of movement of proton from IMS to matrix is known as pumping of the protons from here to here, and this actually leads to the formation of ATP from ADP and PI, which is mediated by the ATP synthase. Huh. Yeah. So yeah. So this is the end of. So we have already discussed the all 10 question of week 1 assignment of the previous course, previous year's course. So this week 1 assignment question has been done. So now, as I already told that we will divide the lecture by assignment solution, and if you have any doubt in your week 1 lecture videos, or if you have any doubt, so you can ask me. So let's go back to the meeting, and yeah. So now the assignment discussion is over. So if anyone have any doubt in the assignment discussion, please tell me, or any doubt throughout this assignment discussion, or actually any doubt in assignment or the lecture video, you can tell me. Any doubt in week 1 assignment or week 1 lecture video, you can tell me. I mean, assignment means the assignment which I discussed here, the previous year assignment that I'm only supposed to discuss with you. So in this assignment discussion, if you have any doubt, you can just please tell me, or if any or throughout the course, if you have any doubt in especially week 1 lecture videos, so you can just tell me. You can ask your doubt now. So will anyone come for asking the doubts? Any doubt you have? So the assignment question which I have discussed, so is it fine, or you need more explanation, or you want to repeat me something again? Just tell me. Huh? Sorry. Okay, thank you. But if you but if anyone can have any doubt, or if you have any doubt regarding your lecture topics of I think you have already seen the week 1 videos. So if you have any doubt, or in the so you can just tell me, or if you have any doubt, you are welcome to ask. Okay. So other people, do you have any doubt? Please ask me if you have any doubt.
Yes,
No, sir. No doubt.
Sorry, you have any doubt?
Sir, can you Can you explain the E3 ligase, the two main E3 ligase, ring and hex?
So you are asking for the mechanism of uh, ethyl ligase, right? The how the
Yes, sir. Yes, sir.
is it
Some elaborations on.
ethyl ubiquitin
Yes, sir.
ligase,
Yes, sir.
right?
Yes, sir. Yes, sir.
Okay, okay. Yeah, so I will just try to discuss about ethyl ubiquitin ligase. So okay, so I will. I'm trying to use this whiteboard. It's I'm also new in this platform, so I'm also facing difficulty here. So I think I can share this whiteboard. Let me just stop sharing my PPT. Yeah, now I'm sharing the whiteboard. Just give me some time. Okay, so just kindly confirm me. Just can you see the whiteboard? All right.
No, sir.
Now,
No.
okay. So now I can do one thing. I will just how to- yeah. So I can just share the screen. Then wait. So my screen is now visible, right?
Yes,
Yes,
sir.
sir.
Okay. So now can you see the whiteboard? Some popping up here. Untitled Google Chrome. Can you see now?
Yes, sir.
Okay. Okay. Fine.
Yes, sir.
Okay. Okay. Fine. Okay. So as you asked for the ethylbutyl ligase ethylbutyl ligase, so as you already know, the main function of ethylbutyl ligase is to add the ubiquitin to its substrate. So can I just ask that what kind of addition is this addition of ubiquitin by ubiquitin ligase? It is covalent or non-covalent?
Yes, sir. Covalent.
Covalent, right?
Carboxypeptide bond.
Covalent, right? And so just tell me one thing: when
Yes,
two
sir.
amino acids are in any protein, when two amino acids are there, conjugated by the peptide bond, so there is already a peptide bond which is covalent. So now in in a protein, when you make another covalent bond with the ubiquitin, that is the thing which happens here, right? When a protein is ubiquitin annihilated, so there is actually another covalent bond between the ubiquitin and the protein. So in addition to the peptide bond, there is another covalent bond which is formed during this ubiquitin addition. So can you just tell me what is the name of this bond, of this covalent bond, which is uh, except this you know peptide bond? What is it called? This covalent bond?
Isopeptide.
Yes, nice. Isopeptide bond. So this is a important point. So now just start. Here I will just try to yeah. So just imagine that this is a it's a ubiquitin ligase enzyme, right? So for its ubiquitin ligase enzyme, it has a binding site for sorry, yeah. So it has a binding site for E2. It has a binding site for E2, and this E2 is, as you know, that this E2 is covalently conjugated to the ubiquitin. So it will be covalently conjugated to the ubiquitin, right? Yes. Yeah. Yeah. So this is ubiquitin. Okay. So this is ubiquitin and this is E2. E2 ubiquitin, and let's just nomenclature also this also E3. E3. Yeah, this is E3. Okay. So this has a binding site of E2, which is covalently conjugate to the ubiquitin. So now the role of this E3 ubiquitin ligase is to transfer this ubiquitin to the substrate from E2. So this E3 ubiquitin ligase has a also a binding site for substrate. This is a substrate binding site. So I will just label it as S, substrate binding site. Yeah, substrate binding site. So now this E3 ubiquitin ligase will mediate the transfer of this ubiquitin from E2 to this substrate. Basically, from E2, yeah, to this substrate. So this role is played by the E3 ubiquitin ligase enzyme, and here the one of the hypothesis of mechanism of the ubiquitin ligase is that it can have the ability to place this ubiquitin and the substrate to the close proximity, and that will facilitate the transfer of the ubiquitin from the E2 to the substrate. And the substrate, as you know, that the substrate, the mainly the ubiquitination site contains which amino acid residue? In which amino acid residue the modification, this modification happens in the substrate?
Lysine.
Lysine, right? Why?
Lysine.
Why lysine? Why lysine? Why not aspartate?
NH2
Why a basic?
terminal.
Huh? Sorry?
NH2 terminal.
Yes, NH2 terminal. In this NH2 group, end has the lone pair of electron, right? So it can donate this lone pair of electron to the CO group of ubiquitin. So this is a ubiquitin which has a wait. Yeah. So this is a ubiquitin. In the C terminal, it has CO group, right? Oh, my writing is horrible. Yeah. So in just C double one O, right? C double one O, because this is the C terminal end, and yeah, so this is the C terminal end. So in this C terminal end, you can see that this carboxylic end, this C is electron deficient, right? Because it is bonded with electronegative oxygen. So it is electron deficient. So it will always will seek for a residue which has lone pair of electron, like a basic residue, as lysine has the nitrogen which has a lone pair of electron. So this lone pair of electron of lysine of this substrate can donate its electron to here, the C terminal residue of the ubiquitin, and they form a covalent bond, which is which is also a CONH bond. It is also a peptide bond, but it is an isopeptide bond because this is not between two because this is not between the N terminal and the C terminal end of the lysine, because it is forming between the C terminal of ubiquitin and and a side chain of the lysine. So that's why it is known as isopeptide bond. So this electron deficient CO group, the C atom will make covalent bond with the NH2 group of the lysine. So in this way, this reaction will happen. And here, the role of the E3 ubiquitin ligase is to facilitate this transfer to make this E2 and this substrate close close proximity so that the transfer can be occurred. And the role of E2 is here. I will go to the upstream events of the E2. So the role of E2 is here that as E3 has the binding site of E2. See, only ubiquitin can't just come and bind to the come and sit to E3, right? Because the binding site of E3, because the E3 has binding site for E2, not for ubiquitin. So that's why E2 is required. First, E2 will be conjugated to the ubiquitin, and then E2 will be sit here in the E3 because E3 has binding site for E2. So due to this help of this E2, this ubiquitin will be in the close proximity to the E3 as well as the substrate. So now this transfer can be occurred, which is mediated by the E3 ubiquitin ligase. And before that, as you know that first E1 will be there. There is a E1. So E1 will first the E1 has its N terminal. So the C terminal. So the C terminal of the ubiquitin will form a peptide bond with E1, and then the ubiquitin will be transferred to the transferred from the E1 to E2 to the NH2 N terminal of the E2. CO of ubiquitin will be uh, will form a covalent bond with the NH2 N terminal of the E2. So now this E2 ubiquitin conjugated complex will be bound to this E3 ubiquitin ligase, and this E3 ubiquitin ligase will finally make the transfer of ubiquitin from this here to the substrate. And after this transfer of the substrate from here to the substrate, I mean from E2 to the substrate. So after this, waits, there's a possibility of formation of addition of more ubiquitin here to form a polyubiquitin chain. This is known as polyubiquitination here, right? And this and ubiquitin itself has many lysine, right? K30, as I can remember, K36, K40, some lysine residues are there, where these lysine residues form a same role which was played by the lysine residues of the substrate, which will donate the electron of the lone pair of electron of the NH2 group, N atom of the NH2 group of the lysine, and it will make a covalent bond with the C terminal of ubiquitin, and this ubiquitin has a C terminal glycine. Okay, and this CO group which you are watching here, this CO group is actually the C terminal end of the glycine, which is the end last residue of the ubiquitin. So this CO group of glycine residue will make a covalent bond with the previous ubiquitin NH2 group. Similarly, last glycine of this ubiquitin will form a covalent bond with the last of the previous lysine of the lysine of the previous ubiquitin. So in this way, this polyubiquitin chain will be formed. So now just tell me one thing, especially this question is open to all, that after this so if we will tag the ubiquitin, I think you guys are aware about the tagging of gene, right? So can you tag the ubiquitin at the C terminal? Can you tag? Can you tag by any epitope, because epitope tagging is there for the Western detection of protein. So can you tag the ubiquitin by C terminal? What do you think? Can anyone want to answer? Hello. Hello. Yeah. So can anyone have any idea? Can we tag ubiquitin
Double
at the
glycine.
huh? Sorry.
It ends with double glycine.
Yeah. 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 See, if we tag the ubiquitin at the C terminal, then this CO group of the glycine will form a covalent bond with the tag, right? So now this C terminal glycine, CO group of the C terminal glycine will not be available to form this covalent bond with the substrate. So the substrate will not be ubiquitinated if we tag the ubiquitin in the C terminus. So that's why ubiquitin is generally tagged at the N terminus, so that this ubiquitination reaction will not be uh, prevented. So each ubiquitin ligase, there are different each ubiquitin ligase. Like there are Hec domain containing each ubiquitin ligase, ring domain containing each ubiquitin ligase, ring domain contains containing each ubiquitin ligase. So for the most common uh, ring domain containing each ubiquitin ligase, the mechanism I have showed you, that the transfer of the ubiquitin will be occurred from E2 to substrate. E2 to substrate. There is no so the ubiquitin will not be bound to enzyme and substrate. It will be directly transferred from the E2 to substrate. So in contrast, the Hec domain containing each ubiquitin ligase, so they has a catalytic cysteine here in the active site. There has a catalytic cysteine. So as you know, the cysteine also has sulfur, right? And sulfur has lone pair of electron. Right or wrong? Sulfur has cysteine has sulfur. Sulfur has lone pair of electron, right?
Yes, sir.
Yes. So yeah. So the ubiquitin ligase, which contains the uh, Hec domain. So in this case, this direct transfer will not be occurred. Instead of that, this E2 ubiquitin will bound to here. Fine. Now from here, the ubiquitin will be form a covalent bond. This same, the CO group of the glycine. This group will form a covalent covalent bond with the sulfur of cysteine of E3. So here the ubiquitin will be covalently bound to the E3 in this intermediate step. This is the intermediate step, and after forming this covalent bond with this E3, it's cysteine basically. So from here, after that, it will be transferred to the substrate lysine. Okay, it will be transferred to the substrate lysine after that, not directly. So this kind of ubiquitin, so enzymes which follows this kind of ubiquitination mechanism, these enzymes are known as ring domain containing its ubiquitin ligase. And there are another, recently they have found that, not recently, it is always there, that ring between ring domain containing its ubiquitin ligase. So that is basically, let me just, yeah, for RBR, which is known as RBR. So in this RBR, can I copy that? Or? Yeah, I can copy that. Yeah. So here, yeah. Is E2, this is ubiquitin, okay, and this is our RBR, ring between ring domain. So basically there are two ring domains, ring between ring. There is ring 1 here, here. There is ring, we can consider it as ring 1, and also here there is ring 2. Okay, let's just put it forward. Yeah, ring 2. So what happened here? Yeah, so here in the ring between ring domain, the yeah, not here. yeah, in the ring between ring domain, first this is also a EC ubiquitin ligase. So first this E2 ubiquitin construct, the same like before. This will bound to the E2 binding site of EC ubiquitin ligase. Okay. After this binding, this ubiquitin, like the Hec domain, so the ubiquitin will first this ubiquitin will be transferred from here, same the CO group of the C-terminal glycine. So it will be transformed from the here to here. This ring one domain has a cysteine, like cysteine which contains the sulfate, and after that this sulfate, yeah, so yeah, and after that this sulfate bound will be transferred to the substrate, and the substrate is, yeah, substrate. And here I think I'm getting confused here because I'm not sure that whether the substrate binds to the ring 2 or not. Okay, that I need to check. So I will so I will check this about this ring 2 and I will tell you later. But the main thing is that there is two ring domain. In one ring domain, like the Hec domain containing ET ubiquitin ligase, the ubiquitin will be transferred here, and from here it will be transferred to the substrate. And the role of this ring 2 domain I will just let you know later. Okay, so this is basically, so mainly focus in this ring domain and Hec domain containing ET ubiquitin ligase. So is it clear now? The mechanism of
Yes,
how.
sir.
So yeah, so do you have any doubt which you posed earlier? Doubts are cleared, or if you have still any doubt regarding this?
No, sir, it's clear.
Okay.
Yes, sir. Yes, sir.
Okay. Nice. Okay.
So it's about
Yeah,
the
so
head.
anyone-
Ma'am didn't,
Sorry?
but the ma'am didn't.
Sorry? Yeah, please continue.
The ma'am didn't consider the cysteine amino acid. So that's why it was problem.
Actually, Hec domain, it has this, if I am currently remember, I think, yeah, it has the cysteine residue.
Ma'am didn't mention the cysteine amino acid.
Okay, I think, yeah, I think it will be definitely mentioned, or the purpose of the video was actually what she did, it was perfect. I think it was about the context of the video. So yeah, yeah, but anyway, you can just put it as your you can just take it as your information, and please go through the lectures, which are very useful for yeah, and very helpful. And yeah, and you can just take it as a example for Hec domain containing its ubiquitin ligase. Okay, so any doubt you have, your doubts are cleared, or anyone have any doubt in this explanation? Can you please let me know? Hello everyone.
Hello, hi sir.
Yeah, yeah.
So I have another doubt regarding the terminology of some like CDC. When you generally say CDC, what do you mean? Like, is it cyclin kinase? Is it cyclins or is it phosphatase? Because like, can you just give a small clarification regarding that?
CDC, basically CDC stands for cell division cycle. So yeah, but if they are found to have any roles according to the function, or according to the, yeah, according basically to the function, they have role, are also used as a cofactor. I mean, the cofactor of many cell cycle proteins. So this, yeah, like this CDC, for example, CDC25, I think is a phosphatase, right? CDC25, am I right?
Yes, it's a phosphatase.
Yeah, so according to the name, according to the function of their yeah, yes, according to the function, you can tell it as phosphatase, but as they have role in the cell division cycle, so according to that, they their nomenclature was found as cell division cycle, CDC. And later the function was found that, yeah, it is a phosphatase, or is a kinase, or it has some different cofactor or yeah, cycling type of thing, or yeah. So basically, if they have role in cell division cycle, uh, to regulate the cell division cycle, to make the transition of phases, or to prevent any uh, the or to prevent the transition of any phases. Okay, so if they have any role in cell division cycle, they are generally, you know, to, named as cell division cycle CTC proteins. Some of them are, as I told you, phosphatase, like CTC25 is a phosphatase. Yeah, this is a function. Okay.
Okay, sir.
Any more doubt?
Sir, what will be the difficulty of the exam?
Difficulty of the exam, that I don't have any authority to say about that, and I don't have any knowledge about that also. I can't predict, right? I can't predict. So the main thing I can tell you, that you have to follow the lectures very, very carefully, and it will be very helpful for you. It is a very informative and very conceptual lecture. It will build your concept. So lectures have to be followed very nicely, and you have to complete your assignments. You have to, yeah. So, and yeah, and also you have to read some books, and especially the lectures. So that is only the, way to get a good score in exams. I am not authorized, or I don't have any idea how we
So is
how.
there any chance we can get a offline internship?
Huh? Sorry.
Is there any chance we can get a offline internship after the exam if we score enough good?
Actually, actually, this live session is not meant for asking some things. If you have doubt in these things, you can just directly contact NPTEL, or official website. This
Okay, sir.
this session is only this session is only designed for, solving your doubts, solving assignments. And if you have any doubt from your lecture courses, as I told you, week one lecture, if you have any doubt, you can tell me, just that I am supposed to do only. Okay. So if you have any doubt, please ask me
Okay, sir. Sorry.
regarding the lectures or anything else. I mean, lectures or any more doubt.
No, sir.
Okay, so can anyone tell me that how, about some information about the DNA double helical structure, which was in your lecture content? DNA double helical structure, and how many types of DNA you know about? Right-handed DNA, left-handed DNA,
A form, B form, and Z
then form.
form.
Yeah, very good. So among them, which one is left-handed? Z
Z
form.
form.
Yes, yes. Any doubt? Where the where the glycolysis is taken place? Okay. Hello.
So glycolysis,
Glycolysis.
it takes place in the
Yeah,
cytoplasm.
cytosol, right? And TCA cycle in mitochondria, right?
Yes, sir.
So why TCA cycle in mitochondria? Why not cytosol? What do you think? What can be the reason?
Sir, I think because after
So
the
because if
pyruvate,
Sorry? Sorry?
Sandeep, please continue. Okay, sir, I was thinking PCA cycle take place in mitochondria because when pyruvate is produced after glycolysis, that pyruvate is converted to acetyl coenzyme A, and that acetyl coenzyme A can enter through that cytosine. Some that is one transportation mechanism in the membrane of the mitochondria, which helps in the transportation of acetyl coenzyme A into the matrix, and
Okay.
that helps in the binding of that TC cycle. What that acid exactly? I can remember that acid. Oxaloacetic acid, right? Oxaloacetic acid, and then it forms the citric acid, the first three carbon compound of the TC cycle.
Yeah. 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 So that can be one reason that, as you told about the transport issue, and acetyl CoA must be formed because otherwise you can't have the high energetic bond, and if you don't have the high energetic bond, you can't have the ATP, right? So acetyl CoA is purposefully formed by the cell, so to just get energy, to get ATP, and yeah, and it can be the reason that the transport of acetyl CoA is possible, so that's why they are shifted to the mitochondria. And another big thing is the all of the enzymes, if you will see the all of the enzymes which are involved in the TCA cycle, right? Right, and at the output of the TCA cycle is FADH2, NADH, FADH2, CO2, then ATP. So all of this output of the ATP, citric acid cycle are occurring in mitochondria, and the enzymes which are mediating this, they are residing in the mitochondrial matrix, right? Right? Am I wrong? All
Yes,
right.
sir. Yes, sir. Yes, sir.
yes,
Yeah.
sir.
So to get access, yeah, to get the access of the TCA cycle enzymes, to get the access, so they have to be transported to the matrix, right? If they will remain in the cytoplasm, then they will not have the access of the enzymes, and the TCA cycle will not be continued, right? So that's why I think this can be, I mean, in addition to your reason, this can be also a additional reason that to get access of the enzymes to continue the TCA cycle, it is required to just you know getting transported to the matrix, right? Yeah.
And sir, I also think that after the production of NADH and FADH, the
FADH2.
electron transport chain also NADH and FADH. Sorry, sir. That was also means the electron transport chain also take place in the means like inner membrane of the
Yes. Yes.
mitochondria.
Yes. Yes. Nice. Very good. This is another reason because the all the complexes which mediates this electron transport chain, because see, if there is no electron transport chain, then there is no ATP, there is no proton pump, there is no ATP, there is no proton gradient, there is no ATP. So to get the so that's why to let the ATP synthesize ATP, this electron transport chain is required, and this electron transport chain is residing in the inner membrane, right? So that's why this process has to be, you know, taken place in matrix, right? Yeah, yeah. And see, there are some mitochondrial matrix proteins, and there are some mitochondrial membrane proteins, right? Like your, as you just told, that the electron transport chain, the complex one, complex two, complex two, complex four, ATPC, they are all membrane proteins, right? And some of the proteins are matrix. They reside in the matrix. So what do you think that how this information is how is the information read by the cell, that I need to fix this protein in membrane, and I need to localize this protein in matrix. So how this decision is made by the cell? How can the cell sense? Any idea? Just see, this is also part of, just hypothesis. We can just presume. So what is your idea that why that what is the sensing process of the cell, that how can cell sense that I need to localize this protein to membrane, I have to localize this protein in the matrix? How can cell sense? Is the information is written in the amino acid sequence, or how? Any idea? Just be open, just be open, just discuss. Any idea?
No, sir, I think due to evolution process, slowly the mitochondrial means the information in the lecture of ma'am, she presented that endo symbiosis hypothesis, where the eukaryotes evolved when prokaryotes combined together. Mitochondria was another prokaryote. Sorry, mitochondria was another prokaryote. Then cyanobacteria, another prokaryote. They combined in another archaea to form a plant cell and mitochondria in animal cells. So slowly in this evolution process, I guess, due to the presence of this information, they just evolved. I think that one reason behind that.
Yeah, that can be a very valid reason. Yeah. I also think so. That can be a very valid reason. And in addition to that, can you think of any reason? Or see, one thing, it can be happen that transmembrane protein. If you just look at the sequence of the transmembrane protein, it has alpha helical transmembrane right? So that is the indication that this proteins has many transmembrane hydrophobic transmembrane alpha helices. So they have to be put in the membrane, because then only it can form a hydrophobic interaction with the membrane, and so that can be a also reason here also it can be a reason, or yeah.
Yes, sir, that can be one good reason.
Okay, any doubt you have? Any more doubt? Someone in the assignment or anything?
Sir, actually, I have one question. Uh, the
Yeah.
DNA have A, B, and Z form of DNA. So I was thinking, what are the forces which are responsible for those different types of DNA? Like for example, if you go for protein, primary, secondary, tertiary, and quaternary structure, the
Hmm.
bonds like hydrogen bond, van der Waals interaction, disulfide bonds, these helps in making the complex structure of protein. But
Uh,
what about the DNA, A, B, and Z form of DNA?
are you talking about the chromosomal organization of the DNA, or DNA double helix? I mean, these hydrogen bonds.
Also the structure means like if you say that A, B, and Z, as far as I remember, in A the structure is little bit similar to B, but whereas in case of Z, it is it's completely flat like because the major groups in case of Z are not present.
Yeah.
And I think the distance between single base pair in case of A is 2.3 Armstrong, whereas in B it is 3.3,
Yeah,
and
B
where
is more
in Z
elongated.
it is,
Yeah.
sir.
Yeah, B is more elongated, right? The,
Yes, one. B is more elongated, but whereas Z is completely like straight, like completely linear structure.
not linear. It is also a double helical structure. It has,
Okay.
but the
So, what helps this DNA to get such structures? Like, which bonds? I'm saying means like the hydrogen bond is the main bond which helps in the formation of double helical structure of the DNA. And beside that, they have a phosphodiester bond which links the nucleotides.
yeah,
Beside these two bonds, is there any other bonds which are present in this DNA structure?
in DNA there is also a bond called base stacking interaction. This is a non-covalent bond. Like unlike the 5 prime to 3 prime phosphodiester bond, this is a covalent bond. Hydrogen bond is also a non-covalent bond, and there is also another kind of interaction is base stacking interaction. Because see, what do you think? What is the nature of the hydrogen bases? They're hydrophilic or hydrophobic? Phobic.
I think, sir, hydrogen bonds are hydrophilic
No,
because
not hydrogen bonds. Not hydrogen bonds. I'm asking about the nitrogen bases.
hydrogen bases.
Nitrogen bases.
Hydrogen. Oh,
Like iron.
nitrogen bases.
Yeah, yeah. Nitrogen bases.
Nitrogen bases. Mm, I think they're hydrophilic.
Nitrogen bases are hydrophobic, I think. Right. That's why, see, that's why in the DNA structure you will see the phosphate groups are projected outside, but the nitrogen bases are projected to the inside, right? Because in the outer side of the DNA there is what? There is polar environment, right? There is more generally aqueous environment, polar environment. So so for that, you need to project a group which is hydrophilic. And in DNA, phosphate group is hydrophilic. That's why phosphate groups are projected outside. Right? That is true for both of the strands. But the nitrogen bases they are projected inside because they are because it is just simple like protein. See, in protein, if you will think about the globular protein, especially, so in this globular protein, the hydrophilic amino acids will face outside, but the hydrophobic amino acid will be faced to the inside. They will be buried inside the core, right? They don't expose the outer polar environment, right? So for that, so similarly here in DNA also the nitrogen bases are are actually hidden from the outside polar environment. So they are projected inside, not outside. See, that's why. So from this indication, you can also know that the nitrogen bases are hydrophobic. So so here is a base. Next nuclear is here. So here is also a base. So now two nitrogen bases are close together. This is nucleotide 1, this is nucleotide 2, and they have two nitrogen bases here. Just imagine. So they have a base stacking interaction, which is a hydrophobic interaction, which is like a hydrophobic interaction because both of these bases are hydrophobic. So they have a hydrophobic interaction, which is known as base stacking interaction, and which plays a role in stability of the DNA. This is another kind of force which we are asking about. This is another kind of force is base taking interaction except the hydrogen bond and the phosphodiester bond. Does it clear your doubt or
Yes, sir. Yes, sir. Okay. Thank you, sir. Thank you so much, sir.
no problem? Okay, and Yeah. So in assignment, let look for the assignment. See in the assignment, yeah. I'm just sharing my screen. In the assignment 6, you can see here. In assignment 6, question 6, you can see that catabolic and anabolic pathway can be coupled in cells because of the answer is this: free energy released by one pathway can drive the other, right? So, so what do you think? The in which form the free energy is being released here? In which form the free energy is being released here? Which form?
So ATP.
Yes, ATP. Yes, ATP. And I think you can be able to know some anabolism pathway where you can see that ATP is consumed to form a macromolecule. So in this way you can see that this free energy is driving to couple this catabolism and catabolic and anabolic pathway. Okay, uh, so in E1, E2, and E3, so can anyone tell me that the role of U1, E2, and especially U1 and E2 in the ubiquitin ligase activity, as we already discussed, the role of U1 and E2 in the ubiquitin ligase activity of E3. What is the role of them, U1 and E2? Can anyone try to? Anyone wants to try?
Yes, sir. I'll try.
Yeah, sure.
So E3 brings the E2 ubiquitin and substrate in close proximity. Then E2 has a binding site for E3.
Yes.
Then first ubiquitin binds to E1 and then it's transferred to E2. Am I right?
First?
Like E1 is also there, right? First ubiquitin
Yeah,
binds
yeah,
to
yeah,
E1,
yeah. U1,
then
U1 is also.
subsequently it binds to E2.
Uh,
Then after that like the CO
who
group
does
of,
bind to E2?
can you repeat
Who,
it, sir?
who does bind to E2? Who is being transformed from U1 to E2?
Ubiquitin.
Ubiquitin, right? Okay.
Yeah,
Huh.
yes.
Huh.
Then finally the like there is the side chain from substrate like the CO
Yeah.
group of ubiquitin will bind
Yes.
to the NH2 group of lysine on the substrate, and
Yeah.
finally ubiquitin will be transferred to the substrate.
NH2 group of which amino acid of substrate or which kind?
Uh, on the substrate it's lysine, and
Yeah.
ubiquitin it's glycine.
Yeah, a CO group. I mean C terminal end of ubiquitin, and ubiquitin has the glycine in the C terminal, right?
Yes.
Yeah. So it has a CO group, right? Because it is
Yes.
a C terminal tail. It is electron deficient. It will form a covalent bond with the NH2 of lysine of the substrate.
Yeah, an isopeptide group.
Yeah, and this bond is known as isopeptide because it is not peptide, isopeptide.
Yes.
It is also a covalent bond, but it is not peptide bond. That's why it is known as isopeptide bond. Now, just tell me among this E1, E2, and E3. So, what do you expect that who should be more abundant and who should be more, less amount in the cell.
I,
Among
th- yeah, I think sir E2 should be more like E2 should be more. I don't know
E2 should
because
be most abundant. You are saying that?
I think I don't. I'm not sure because
Okay,
like
just try, just try.
yeah,
What is the reason for thinking that?
because like ubiquitin will bind to E2.
Yes.
So and I from the figure I know I said think that E3 is somehow big, so it's like substrate for everything will happen. So one more than one of these things can happen on a single E3. Like I'm thinking like that. I'm not sure.
Actually, you are pretty close because see the most abundant is E3, and the
Oh,
reason is to make the variability because see each E3 has different substrate, right? This substrate, if your cell has plenty of like 56, 50, 51, like plenty of E3, then each E3 has separate substrate. Okay, so if there was only one E3 in your cell, then every substrate which are known to be ubiquitin related, so every substrate could be modified by the E3, right?
yes.
But that is not we want because we want here regulation, right? Each subset we just don't want to get every time modified, right? So for to make the variation and to bring the regulation, that's why there is multiple E3. So each substrate, suppose just think that this substrate can only be modified by this particular E3. Okay. So if this is the case, then there will be many E3. So if the substrate is only modified by E3, then the substrate will only be modified if there is expression of this E3. And you know that gene expression of E3 or any gene expression can be regulated in eukaryote, right? So when there is a requirement of modifying S, I mean to ubiquitinating S substrate, then only this cognate E3 will be. That's why it is known as cognate. That then only this cognate E3 will be expressed, and then only this substrate will be modified. Not
Okay.
every time. So that is the regulation here taking place. See, the most downstream is among E1, E2, E3 is E3, right? E3 is the most downstream. So that's why cell has, I mean, the nature has designed it in such a way that E3, there is multiple E3, so that each substrate has different cognate E3. It is you can just think of a purpose of the regulation. Okay.
Okay, sir.
And after that, E2 is less abundant than E3, but more abundant in E1. Right. And yeah, so it is generally I'm talking about the context of the budding yeast, and many animals, I mean, many species, I think, follow this. So in budding yeast, there is most abundant is E3. Then, uh, then after that, E2 comes, and there is a very few. I think there is only one U1 in budding yeast, so that it is. So the purpose of the save energy. See, if there is one U1, so one U1 can do this E2 ubiquitin, right? So one U1 is enough to modify this E2. So now there is several E2 and now there is more number of E3. So here the energy consumption was taken place, and here more number of E3 will bring the substrate specificity, actually substrate specificity for the regulation of the utilization. That's why E1 U1 is the least abundant, E1 is the least abundant, then E2, then E3. E3 is the most abundant, E2 is lies in the medium, I mean the mid region, and then E1. E1 is the least abundant. Okay, so that is the E1, E2, E3 concept. I mean the abundance abundance scenario inside the cell. Okay. Uh, So please, some people are also joining. So if the people who just joined later, so do you guys want to repeat me the assignment once again? The solution of the assignment. Can you please let me know by speaking? The guys who joined later, do you want to discuss the assignment again? Who missed the discussion? Can you just Let me know. Should I discuss it again? Okay, some people I noticed that some people have just joined, so I just want to quickly revise the assignment again because they have missed. Yeah. So this, yeah. So this is the first question was there. Which of the following is general characteristic feature of the prokaryote? There are four options, and the option is of course the nucleoid, because nucleoid is only the thing which is unique to the prokaryote, right? No mitochondria. Yeah, nucleoid is the unique thing from the prokaryote. And this is the, I just discussed about the nucleoid, which is a nuclear genomic DNA, the organization of E. coli. So here there are some portion of genomic DNA, non-structural left, non-structural right, left, original replication, termination. So they are, if you will follow the color, you can see that here, so they are organized in such a way. This region, green region here, termination region here, left here, right here, non-structural left here, non-structural right here, original replication, this region here. That's why they are organized, and this organized structure of nucleus is known as nucleoid. And they, in contrast to the nucleus of the eukaryotic cell, they are not surrounded by the nuclear membrane. Second question was the formation of the acidyl. So yeah, so second question is the formation of acidyl CoA in which region of the mitochondria? Answer is the matrix. So you can see here from this diagram. So in the cytosol, you can see the glycolysis happens from glucose to pyruvate, and pyruvate will enter to the matrix. In the matrix, the PDH, pyruvate dehydrogenase complex, will lead the formation of acetyl CoA from the pyruvate, which is a decarboxylation reaction when one molecule of CO2 will be released. So this reaction is taken place in the matrix. Third question was the experiment performed by Avery, McLeod, and McCarthy. The transforming principle was identified as, the answer is DNA, because uh, when they yeah, so in the only case of DNA, when they are self-free extract contain DNA, the A strand, the virulent strand, A strand self-free extract, when the A strand self-free extract contain the DNA, then only this then only they were able to make this non-virulent R strand to virulent A strand. So this transformation was occurred only in the presence of DNA, not in the presence of protein, presence of RNA, presence of carbohydrate, presence of lipid. It was not occurred. Only in the presence of DNA it was occurred. That's why they have decided that the transforming principle is DNA, not other things. Which of the following organelles does not contain DNA? Nucleus has, we already know. Mitochondria has DNA, mitochondrial DNA. Chloroplast also has DNA. So the answer is lysosome. And in lysosome, we just discussed briefly about lysosome. It contains the acid hydrolase, which are active at acidic pH about 5, which is maintained only in the lysosome but not in the cytosol, which has a neutral pH. So, and lysosome is a membrane enclosed organelle containing enzymes capable of breaking down macromolecules like lipid, protein, nucleic acid, carbohydrate, and to maintain their acidic neutral pH inside the lysosome must actively concentrate proton. This is accomplished by a proton pump in the lysosomal membrane, which actively transports proton into the lysosome from the cytosol. Fifth question was, which of the following combination makes a nucleotide? So you can obviously see from here the sugar, nitrogen base, and phosphate groups. They are they are actually building block of nucleotide. And here I just put some images from here, which are deoxyribonucleotide, because in the 2' group they have H. It is deoxyribo, deoxyadenyl,